In this video we will discuss about the effect of PSC derived Galectin-1 on the PANC-1 cells, whether they induce EMT or not. So what I did in order to check this PSC derived Galectin-1's impact on the PANC-1 cells, they co-cultured this PSC, and in the previous video we have understood they have developed different cell lines of the PSC, that is one in which Galectin-1 is overexpressed, one in which Galectin-1 is knocked down. So when they co-cultured this different kind of PSCs with the PANC-1 cells and then check the morphology of the cells, what they are able to find out is that in the case of wild type and in the case where Galectin-1 is knocked down, there is not much change in the morphology. But for the case where PANC-1 is overexpressed with PANC-1 is co-cultured with PSC in which Galectin-1 is overexpressed, in that case they are able to see that PANC-1 cells are tends to elongated, and more prominently they are tends to grow individually. So this shows one of the key difference in the PANC-1 cells in the conditions where Galectin-1 is overexpressed. And next what they did is they did the Western blot analysis to check these markers for the EMT, and at the same time they also check the prominent signaling pathways that triggers this EMT, that is NF-kappa beta P65. And for the markers of the epithelials behavior they used E-cadherins, and for the markers of mesenchymal's behavior they use Vimentin, and simultaneously they also check this MMP9 expressions. So this is the control, this is the Galectin-1s, which shows that the first column is of the Galectin-1 knockdown, second one is the Galectin-1 wild type, and third one is the Galectin-1 overexpressed. And in the case these MMP9s are upregulated in the case where Galectin-1 is overexpressed compared to the case where Galectin-1 is knocked down. And when it comes to the E-cadherin levels are also high or increased when the Galectin-1 is knocked down, whereas it is decreased when the Galectin-1 is overexpressed compared to the wild type. And reverse observations has been observed for Vimentin, that is Vimentin levels are low when the Galectin-1 is knocked down, but they are high when the Galectin-1 is overexpressed compared to the wild type. And the changes to this EMT is also supported. Changes to this high Vimentins in the Galectin-1 overexpressed is also supported by upregulations of this NF-kappa beta P65 compared to the wild type, and it is low in which Galectin-1 is knocked down. This indicates that the mesenchymal type behavior are increased, and one of the reasons could be upregulations of the NF-kappa beta P65, and also this twist pathway, which is upregulated in the overexpressed Galectin-1's co-cultured PANC-1 cells, whereas it is downregulated in the Galectin-1 knockdown PSC co-cultured cells. And for the control they use tubulin. And once they find out the behaviors, that is PANC-1 cells which are co-cultured with PSC in which Galectin-1 is overexpressed has more EMT changes. So in order to validate further, they did the immunostaining, and in the immunostaining they are able to see that in the case where Galectin-1 is knocked down, they see more signals for the E-cadherins. The red fluorescence denotes the E-cadherin signals. You can see that E-cadherin signals increased when the Galectin-1 is knocked down compared to the wild type. And bottom one is with the hoist or the nuclei. And for the case where PANC-1 is co-cultured with PSC in which Galectin-1 is overexpressed, they are able to see more Vimentin signals compared to the E-cadherins, where or we can say E-cadherin signals goes down and Vimentin signal goes up, which is denoted by green fluorescence, and the bottom image is with the hoist or with the nucleus. So all this indicates, both the Western blot results and the immunostaining results indicates that the Galectin1s, PSC derived Galectin1s might induce EMT changes in the PANC-1 cells. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.